It's Michael. Good morning, everyone. So for today's lesson, we continue. Okay. So after the embryo development in seeds, so the last part will be the mechanism of seed germination, right? Okay. So germination is defined as the emergence of the radicle, which is the first root through the seed coat. Okay. Now, seed germination begins when a seed absorbs water, right? And seed metabolism resumes. Okay. So during the germination of grain seeds such as barley, right, nutrients are mobilized by gibberellins. If you recall, gibberellins is one of the many hormones produced by plants. Now, the mechanism, okay, of the seed germination, right, must always begins when the seed imbibe or absorbs water. So the seed must imbibe water, and this will cause the seed coat to soften, right? And then after the seed imbibes the water, the seed will expand in size, and this will cause the seed coat to rupture. Okay. Now again, immediately after the seed imbibes water, what happens? The embryo will release gibberellin. Now, if you recall, this is the seed, right? This is the endosperm. This endosperm is surrounded by these aileron tissues, right? These aileron tissues consist of aileron cells. Now, we have these aileron tissues, the endosperm. This is the cotyledon, right? And this is the embryo, right? This is the embryo, the seed's embryo, right? So after the seed imbibes water, all right, this embryo will release gibberellin, okay? And then this gibberellin will diffuse. You can see this embryo release the gibberellin, and this gibberellin diffuse into the aileron, right? Now, after the gibberellin already diffused into this aileron, right, the gibberellin will bind with the gibberellin receptor inside the aileron cell. Okay, and then the aileron will synthesize and secretes digestive enzymes, okay, that will hydrolyze nutrients stored in the endosperm. Now, one example of digestive enzyme secreted by this aileron, right, is the alpha amylase. This alpha amylase will hydrolyze starch inside this endosperm into sugar. Okay, other digestive enzymes, all right, synthesized and secreted by this aileron is the lipase and protease. Lipase hydrolyzes fats and oil into fatty acids and glycerol, right, while the protease will hydrolyze protein to amino acids. Now, this sugar from the hydrolyzation of the starch, right, and other nutrients from this endosperm are absorbed by the cotyledon or scutellum, right? And then all these nutrients are consumed by this embryo for the growth of this embryo into seedling. So radicle, right, will emerge first. So this is the radicle, right, emerges first from the seed, followed by shoot. So that is the mechanism of seed germination. Now, the external signals and conditions that trigger seed germination, right? First is water availability. You can't write water in the exam. If you just write water, how does the water, in what terms of the water that can trigger the seed germination? So you can write there water availability. So this water is required to resume the seed metabolism, right, as well as to soften the seed coat. Number 2, oxygen availability, which is required by the embryo for cellular respiration. and growth, because the embryo are living things, so it needs oxygen. Then the third factor is the suitable temperature, right, where different type of seeds have different range of optimum temperature, right, and this range from 5 degrees Celsius to 30 degrees Celsius. This temperature is essential for the enzymatic activities in the aileron, right? It's aileron. It synthesizes, aileron synthesizes and secretes enzyme, and this enzyme, as we have learned earlier, okay, these enzymes hydrolyze the endosperm, and for these enzymes to hydrolyze the endosperm, it needs suitable temperature. Then next is suitable light intensity, where red light, right, will promote seed germination, while blue light will inhibit the seed germination, right. The fifth factor is the concentration of abscisic acid, okay, in the seeds. Lower concentration of abscisic acid promotes seed germination, okay, and while higher concentration of abscisic acid will inhibit seed germination, right. And then the sixth factor is seed scarification, right, and this helps to weaken the seed coat and helps moisture to enter into the seeds, okay. So this will prepare the seeds for the germination process. All right, this the seed structure, as you can see from here, right. This is a common garden bean, okay, or an eudicot that has thick cotyledons. So this is the structure. So eudicot that has thick cotyledons, you can see how the cotyledons are being arranged, right. This is the radicle, okay, radicle that will emerge as root. This is the hypocotyl, and this is the epicotyl. Okay, now cotyledon with thin, eudicot with thin cotyledons. This is how it look like. All right. So you can see this is the radicle, this is the hypocotyl, and this is the epicotyl, right? And these are the cotyledons with the endosperm. You can see it's this eudicot with thin cotyledons are very different with the eudicot with thick cotyledons. All right. So you must be able to identify the location of the epicotyl, hypocotyl, right, as well as the radicle. Now this is a monocot, okay. So monocot, yes, only one cotyledon. This is the cotyledon, right, cotyledon, and then this is the endosperm, this is the coleoptile, epicotyl, hypocotyl, and radicle, right. Please know this structure. Right, now the last part for today, types of seed germination based on shoot emergence pattern. So first we have epigeal germination. Now in this epigeal germination, what happens? A hook forms in the hypocotyl. This is the hypocotyl, right? This. So a hook forms in the hypocotyl, okay, and growth process will push this hook above the ground. You can see how this hook is being pushed above the ground, right? And then this hook will straightens, okay, you can see the hook straightens here and pulls the cotyledon out from the soil. So the epicotyl spreads its first true leaves, okay, you see the epicotyl here where it spreads its first true leaves, and then these leaves will expand, become green, and begin making food by photosynthesis. So this is epigeal germination, right, where it involves the formation of a hook in the hypocotyl. Okay, and now the other one type of germination is the hypogeal germination, which is being exhibited by maize and other grasses, okay, as well as plants that has single cotyledon. So in this hypogeal germination, what happens? The epicotyl or coleoptile, okay, you can see the coleoptile here, it will grow very fast and emerges out from the seed. Okay, next the shoot grows straight up through the tube of the coleoptile. So the first leaf will unrolls, okay, within and carries out photosynthesis. So that's about seed germination, right? So that's done for this chapter. So next chapter will be homeostasis. Okay, bye.